hacer posible, así que gracias aquí. Yo creo que va a ser muy interesante, así que vosotros podéis contárselo a los demás. Así que sin más dilación vamos a continuar con SLAVICA. Nos va a contar su experiencia, cómo se hace pruebas y más en granjas marinas, que las condiciones de control son difíciles. Así que
as I told you in the first presentation, I'm working in veterinary service of CROMARIS, but veterinary service is under the Department of Research and Development. So lots of trials which we are doing in the company is done by my team. In my team there are 3 veterinarians and 3 veterinary technicians, and also we are doing with other departments when we need to make the trials. In company is young, but it's established on the much older companies like San Marimar Mirna and Maricultura Istra, and it's in Zadar county. We have 3 farms near the Zadar. It's in the central part of the coast. You can see here we have 3 farms and 1 hatchery, and on the north of the coast we have 2 farms of sea bass and sea bream. Also we have hatchery which is very close to the NIM. So all the team is working on all 4 or 5 sites. Thank you. There is a lot of trials in the company, and I will do just example of 2 trials which we done in the last 5 years. The need for trials is more and more, so we have a huge demand to make feed trials, and also to test some feed which is, for example, antiparasitic additives. Okay, yes, I can. To be a little bit easier. So I will show you what is the problem with the trials. We have lots of cages, so when we are speaking about the possibility to make trials, we can say that we have more than 300 cages to make a trial, but it's quite difficult to make trial if the origin of the fish size when you're starting with the trial is not same, because you need to test the difference between the cages. And other thing is that it's very hard to take a sample, the correct sample size and correct sample from the big cages. So usually we make our trials on 1 small platform which is made especially for the trials. It is very good because we can make in year we can make, I don't know, 10 trials on this platform. We have possibility to use 30 cages for the trials, and during the year we are changing the trial. How we are choosing what we will do, usually it's on the demand of some department. In this case, when we are controlling the imbibition of the bile colors on the fillet, it was demand for the sales and quality control department, because it was some complaints on the customer on the yellow-green color of the wall of abdomen in the fish. This is something which is quite usual for people who are working in this industry, but the quantity of the fish which is going on the market provoke that we need to have a higher quality of the product, and the complaints like that is something that we need to resolve. The other thing is that in the past we sell a lot of fish as a whole fish, and now we are selling fillets, and in the fillets, when you make a fillet, there is big demand to have a white without any colors which are strange. The first hypothesis for this color was that is depending on the length of starvation, because the fish which is prepared for the harvest is starved at minimum 3 days, depending on the temperature. But now we have lots of fish in the big cages, so sometimes starvation period is more than 3 days. Usually we have at least 15 days of starvation of the fish which is prepared for harvesting. Also, hypothesis was that maybe it's a problem with the food, because we know that in the last 10-15 years the fish feed changed dramatically, so we have a lot of plant material in the feed, and one of the hypotheses was that fish is producing more bile and more salt, and that this is a problem which is appearing like that in sea bass and sea bream. Pictures that in the sea bass this coloring is less than in the sea bream, and also it was a question to resolve why the sea bream has bigger pigmentation. To make the trial, usually you need to make some preliminary trials just to see what will influence on the trial, and we did it in our processing plant. The problem when you are doing trials which are directly involved in the production, that you cannot control the production. Production is our primary goal, and we cannot change anything with the production. And we did it in processing unit, sampling 200 sea bream and 250 sea bass, and we check everything what was possible in that moment. We put the temperature, the pigmentation. This is the pigmentation of the muscle, days of starvation, checking the condition of the fish, liver bile content, the weight of the bile, and problems was that even it was a big sample, the origin of the fish which we took on the trial was from different origin. Also, condition of the fish was different depending on the farm. The food farm used was different, sites and sea temperature on the site, because on the north we have a much colder temperature than on the south. So we made the new plan for the trial, in which the feeding regime will be same for all the cages. We will simulate suspension of feeding and harvesting method as in the production, and we will check it on the trial platform. In this preliminary trial, we give some information which was also useful for the new trial, and we find out that probably the bigger pigmentation in the sea bream was due to the bigger gallbladder than in the sea bass. In sea bass, it was a matter of anatomy of the fish. In sea bass, it's round, small, and the contact surface with the muscle is smaller. In the sea bream, the contact with the gallbladder was very big, because the gallbladder is long. You can see difference in the length of the gallbladder. Other thing that the liver of the sea bass is somehow protecting the gallbladder from the contact with the muscle. Also, we saw how it looks like when the gallbladder is empty, what is going on with starvation. So total amount of the produced gall is greater than it can be spent, and mucus of membrane continuously absorbs water, causing the main bile elements to concentrate. So you can see in this picture how the pigmentation is going big high. And then again the place of the gallbladder is different. So if it's very close to the muscle, the pigmentation will be bigger. We made the two trials for this problem. One was on sea bass 2015 and one on sea bream 2016. With sea bass we made 4 cycles of research which were on the different temperatures. Lowest was on 30.3, so the very low temperature, which is corresponding with our lower temperature in production, and the highest was 25.4, which is also corresponding for our highest temperature in the sea. We made one trial group without repetition, and we tried to make a correlation with the temperature and the length of starvation for the period. Fish is fed with commercial food. Cycle was 10 days long. The day before starts with each cycle, cages were fed to satiation and then starved for 10 days. In these 10 days, samples were taken 6, 12 and 24 hours first day, and every day after that once to see the difference. The control cages were fed daily without starvation and sampled on the 3rd, 4th and 10th days to have some comparison with the results. In the sea bream, in generation 2016, it was just one cycle of research to confirm the first results and to see is any difference between the species, and temperature which we choose was 70.7, something in the middle. It's our average year temperature for Croatia. We made 2 experimental groups without repetition, but we choose to make also comparison between the commercial feed and the feed which we call CROMARIS recipe, and it has a little bit more fish meal and fish oil than in commercial feed. Study cycle was similar like in the first trial, 10 days. The day before all are fed to saturation. After that start for 10 days. Control cages were eating every day. The results which we get from these trials were statistically analyzed to determine whether there are statistically significant differences in the appearance of muscle tissue coloring between the experimental groups, and correlation were made to the length of starvation period, temperature, time from harvest to the processing, the gallbladder weight, liver weight, time of storage on the ice in different food composition. The coloring of muscular tissue, while bile colors, was more pronounced in the fish that were starved, which was the first hypothesis. The gallbladder fills and becomes more heavy in starving fish, while the liver mass is reduced. So it's more space for the bladder to be in the contact with the body wall. The condition index, which is statistically significantly lower in the starved fish, favors the contact of the gallbladder and other organs. Reduced body weight decreases the amount of perivascular fat tissue, and it also was somehow prevention to contact gallbladder with the muscle tissue. Results was also very interesting regarding the temperature, because the first of the hypothesis was that in the cold period, like a winter, we have more complaints on the coloring, and that the sea temperature will affect the coloration of the fillet. But there is no difference in the imbibition with respect to the sea temperature. It was not visible in this analysis. The sea temperature was associated with other observed variables like condition index, lower liver mass, smaller mass of the liver, smaller gallbladder mass, and higher index, which we call CHI, and it corresponds mass of the gallbladder to the body. So lower sea temperature is associated with higher condition index, higher HSI, and higher liver mass. And finally, this was our results. That longer starvation before is associated with more frequent and more pronounced appearance of the coloration issue. And this graphic is the most important variables that work together. The higher the value of starvation period and the longer time it takes for the fish, it will be more frequent the coloration of the fillet. So our suggestion to the processing plant and to production was that if they are planning to make a fillets of this fish, that they need to make it after a few days of starvation, and also to avoid the taking the fish from the storage and making the fillet, because sometimes fish is waiting for the filleting. So if it's possible, it's better to go on the filleting immediately after harvesting, which was not the practice in the last years. So this is example how we can get the very clear and good results if we are making it on the trial, on the trial platform. These results we could also get observing the fish in the processing plant, but it will take lots of samples and time to get the same results. As this industry is very young, and the feed which we are using is not stable, the content of the feed is not stable, we are doing a lot of job with the feed trials. These feed trials we start with, I think 5 years ago, and it's carried out at the production and quality control office request, because the production unit thought that some difference in the results in the farms are in the relation with the feed companies. So we need to make the trials in which every year from that we are controlling the supplier of the feed. So when we decide which supplier we will work with this year, we made the trials and all of them are involved in the trial. Also, we can do it in the farms, but big problem is that it's very hard to find 10 or 16 cages which will be of the same origin, same condition, to be controlled by the food, farming sites, and sea temperature. So now we have this platform, and for each of the producer and each feed we are using, we are making 2 cages for them. The condition in the cages are meant to be very similar to the production. So when we sell fish from these cages, the density of the fish in the cages must be the same as in the production sites. The fish which are going to be tested are tested statistically before the trial to see that is any difference on the beginning. And each month we are doing sampling which is a minimum of 100 pieces with anesthetic, so we can put them again in the cages. This is example of the trial which was on the generation Sibrim 2017. So we have two producers which were compared to also three producers, but on the different recipe of the feed. You can see that this is commercial feed, and down is our CROMARIS recipe. These two producers also can check their fish each month if they want. So the trial is open. If they don't believe or don't want our data, they can come and make it by themselves. As you can see, the results are very similar, which is in the last 4 years we have same or very similar results, but when we make analysis there is no statistically significant difference between cages, saying that the feed companies having the very similar recipe for the same species. And if you have the good feeding protocol, you will obtain the same results. So it's something that we are giving also to our production to see that any difference in their farms is not corresponding to the problems with the producer. And in the CROMARIS recipe, also all three producers give us very similar FCR and very similar growth rate. So we are assuming that there is no problem with our recipe also. The FCR is a little bit lower, but the growth performance is better. In the case of the sea bass, we are also doing the same way, working with 3 different producers, and results are very similar to the results of the sea bream. This is a place where we are doing our trials. It's near the farm Lamiana. So usually we can also compare our trial results with the farm results. Thank you for your attention.
Next time we can go to visit you, because I think it's a nice place to go. So it's summertime maybe. We make this seminar, a workshop, doing trials there. Okay. ¿Alguna pregunta o queréis al final, después de ver la siguiente charla, mejor? Okay. Pues aquí tenéis el título. Cambiamos un poquito de temática, pero vamos a ver otras aplicaciones, y esta enlaza más con la biotecnología azul, de las aplicaciones de lo que puede ser un problema en las granjas de peces. Si tiene valor económico, seguro que ayudará a resolverlo, porque si podemos sacar algún provecho, habrá alguien que se interese por ayudar a las granjas a no tener este problema y por tanto a solucionarlo. Así que, Anne.
Gracias otra vez. So this is a little bit off the normal aquaculture route that we'd been plotting today with the other talks. But I mentioned to Geronimo that I could include this, and he said, yeah, let's put that in as another aspect of novel blue technology. So part of this, I'm going to cover what the definition of venoms are. I'm sure we all kind of know, but there's some divisions in there that it would be good to make clear. I'll talk about how we can go about evaluating venoms, and I'll talk about some of the success stories that have happened historically in terms of turning venom into drugs. Not fun drugs, but useful drugs. And then I'll talk about why marine venoms, and in terms of the Blue Bio-ecnonomy, are probably the next big thing, and how we're going to go about accomplishing that. So let's first just draw some definitions. Venomous versus poisonous. With poisonous animals, you've got to eat it or absorb it. So here's a chap dead after eating a frog, and here's a chap dead after being bitten. So here he's got to bite the frog, here the animal's got to bite you. So a venom must be delivered via a target structure, or delivered to a target via a structure, such as teeth or a nematocyst or other venom systems with glands and dentition and what have you. With a poison, it's passively absorbed, again by eating or through the skin. So some of those systems, the combination of a parotid venom gland and grooved or hollow fangs in snakes, or a venom bulb and a proboscis in cone snails, or nematocysts in jellyfish, versus a poison arrow frog, or versus a fugu pufferfish, both of which you need to eat in order to suffer the consequences. And why does this matter? Why do we care about the distinction? To a point we don't, except insofar as we are going to evaluate venoms for candidates for drugs. So this is technically correct man who's being very helpful to a victim who's been envenomed by a snake here, and he died. So technically correct or not, it does matter to us. And that part of that has to do with the ecological context of venom. One immediate ecological way of dividing up venoms is to look at predatory venoms versus defensive venoms. So defensive venoms would be, for example, those used by bees. And if anyone's ever been stung by a bee or wasp, you know that it's quite painful. And that is by design, because if a bee colony wishes to defend against a predator, if it can't kill that predator, or even better, if that predator is social, it would be advantageous to the hive for that predator to remember and associate bees with pain. So defensive venoms tend to be associated with pain-causing compounds, so that the predator or threat in any capacity will remember the experience and avoid it in future. And if the predator goes on to communicate that information to its conspecifics, then so much the better. And that's likewise with venomous fish, such as stonefish. They're usually benthic or demersal fish, subject to predators. So their venom is quite painful, but generally not lethal. So when you hear about people dying of a stonefish sting, it's usually due to drowning or another secondary cause that's come in because of the pain they're in. Other defensive venoms might include stinging ants or certain wasp species that don't use their stings to hunt, but just use them defensively. And to draw the contrast with that, you would look at venoms that are then primarily used to capture prey. So jellyfish and snakes, primary examples, they use their venom to immobilize prey, and in the case of snakes, they also use it to begin its digestive breakdown before they consume it. You should note that predatory venoms are very often used in defense, but their primary evolution is fairly obviously for purpose of prey capture. A neat example of a crossover venom, so both of these venoms, of course, can be used defensively. And this guy here, this is called an assassin bug, and I've kept him a bit separate because he has venoms evolved for both purposes. So he has a primary predatory venom and a venom delivery system, but a distinct and separate toxin cocktail that he delivers when he's under threat. And that's associated with quite a lot of pain. When it's hunting, it delivers venom that paralyzes and digests other insects. When it's defending itself against its own predators, it's very, very painful, and it comes from a different set of glands. Another example of that is with various types of cobras. So some cobras can spit, some cobras can't. Many cobras can also rear up and form that typical hood behavior. They'll do a threat display with the hood spread. Only some cobras have that ability, and the hooding display is very closely associated with those that have very painful venom, as is with the ability to develop that hood and to spit. So it turns out that the ones that can spit are associated with having quite a lot of tissue destructive materials in the venom. So this causes pain to the eyes. It does not need to be injected in order for it to cause damage. So if you are a cobra that spends a lot of time in an environment where you might be picked off by a bird or a predatory mammal, it pays to develop a threat display with some kind of follow-through. So you don't just put a display on to show that you're frightening, you also then cause pain as a result of that threat display. So these can spit painfully into the eyes. The aggressor will remember it. The aggressor will avoid trying to eat that snake in future. And the types of toxins found in those venoms is very distinct from the types of toxins found in those that did not do that hooding and threat display and the spitting. So the reason I've just specified not king cobras here is that they are in a separate taxonomic clade despite the name, just FYI. Similar drivers of venom delivery systems and venom contents can be seen in box jellyfish. So here its habitat, its mode of predation, its metabolic requirements, and its life stage all kind of converge to determine what its venom's content and its venom's system is like. So as a small jellyfish, it eats shrimps. It has very few large venom-containing cells that can deliver large doses, and its venom has a much lower potency to vertebrates. As it gets bigger, it begins to feed on fish. It reaches sexual maturity, and that nematocyst complement starts to become much larger. So it'll have larger volumes nematocysts that contain more venom, and that venom will become more potent. So when you're initially assessing a venom for therapeutic and potential research, it's a good idea to take that kind of ecological consideration under advisement. So what kind of venom are we dealing with? What does the animal use it for? What's their target? How broad is that category that you're discussing? And what's its mode of capturing prey? Is it going to ambush it? Will it chase it? Does the environment mean that it needs to pre-digest its prey? For example, certain rattlesnakes that live in colder environments benefit from having a greater degree of dermonecrotic and tissue-lytic venoms, so that when they do swallow prey, it breaks down quickly enough in their cold-bodied environment that they won't then have putrefaction happening inside their own stomachs. So all of these factors will affect a given species venom, and even within those factors, take care for geography and life stage within the same species. And then the next thing to look for is clues from clinical envenoming, which would give you a notion of how to proceed in terms of what compounds to investigate. Are the key venom outcomes hematological? Are they in terms of developing hypotension or hypertension? Are they neurotoxic? Are they neuromuscular? Do you see a lot of tissue destruction? And bearing in mind that these occur because multiple toxins are occurring and working all together in concert. So you may not get a clear picture of what's happening based on outcomes such as this, but you can get a notion of what might be going on before you begin to break them down and break them apart. So next step is get yourself some venom, and this can be complicated. Not all venoms are as easy to get as a snake. So a snake is obvious. You get a snake, you make it bite a jar. Carry on. With stuff like jellyfish, there is a difference between in the toxin complement of what you might get from a venom obtained by different means. So the classical way of obtaining jellyfish venom was similar to snakes, so it was so-called milked. And scientists would take clipped tentacles of a jellyfish, lay them across an amniotic membrane that's been stretched over a jar. So you have your jar, your membrane, and put the jellyfish on top of that. You stimulate it with some electrical current, which causes the nematocysts to fire. They fire through the membrane, and you can collect the material that's coming out on the other side of the membrane. However, this introduces contamination from the membrane itself, because you need something so thin as amnion to be able to work with that, but obviously an amnion is itself covered with active proteins. So whether that is a representative venom was very much open to question. Other ways to do it include collecting nematocysts and breaking them up using a sonicator, but then it turns out that tentacle material, even in the absence of nematocysts, is itself toxic. So what do? We ask many questions. So sometimes venom's biology history is just full of people who, in fact, died trying to catch the right venom to create an antivenom for it. And that's where making sure that you have the right animal and the right way of collecting it can be very important, as small amounts of venom are difficult, if not pointless, to work with. Right. So next, you've sourced your venom. Let's say that you've got a riddle or you've figured out a novel technology that can get it out of a jelly. What do you do now? The first step is to look at what it does crudely. So in this case, and depending on your research goals, you might want to look at the whole animal response first. So in this case, you'd be looking at what it does to specific tissue. After that, or within that context, you depending on your goals, to choose the taxon of that model animal very carefully. So if we're studying how a fish-eating cone snail operates with its venom in a human, that is clinically relevant, but is it ecologically relevant? If you then want to find out how that cone snail is going about getting its food and what the ecological advantages of its venom is in obtaining that food, you would want to choose a fish, not a person. Or not a mammal, excuse me. I don't think anyone's testing venoms in humans these days. Anyway, the next thing you'd want to do, once you have enough clues based on that whole animal model, is to choose a target tissue, so that the key cells that you think are being involved in some of these venom outcomes that you're interested in, you'd want to look at how this works in an isolated target tissue, such as a Langendorf heart preparation or extracted chick neck muscle tissue, or in cell culture itself. And that will give you a much better idea of what's happening with the active site biochemistry of the crude venom. In all of those situations, the way you want to kind of gather your data and plot it is to look at dose response. So this particular dose response has to do with the dose leading to a specific time that it takes to cause death. But the y-axis can be anything you want. You don't have to say, I'm studying a fatal venom, therefore I'm going to make this fatalities. You might say, what venom dose is required to slow a heart rate? What venom dose and how long does it take to take that action? What venom dose is required to cause 50% of tissue lysis in a cell culture? All of those are perfectly good endpoints for measuring this. And that is data that you want to file away for when you begin to break down your venom. So most venoms tend to be a cocktail of proteins. Sometimes there are a few lipotoxins out there and some organic acids that are involved, but for the most part, most folks studying venom compounds will be looking at proteins or protein complexes. And the proportion of those depends on the type of venom you're dealing with. It depends on the age of the animal you've gotten the venom from, all of those same factors that I mentioned before. In this case, as a cautionary tale, I've put a coral snake here, since it turns out that depending on where you get coral snakes from, they will display very, very different venom fractions. So coral snakes from Texas might be very, very like high contrast to coral snakes in Florida. So that's something to bear in mind when sourcing So then you want to start separating your venom components. So chromatography comes into it, various chromatographies based on what is actually in the venom. Some of these work better than others. Some venoms are notoriously difficult to deal with because of the degree to which the proteins will adhere or dissociate and then adhere again at the wrong time. So the purity of venom that you're working with does become a consideration once again with this. Once you've decided exactly which proteins or peptides you want to deal with, immunoprecipitation is a really good gold standard for getting something quite pure out the other side of your chromatography procedure. Once again, this is just to show the differences in venom makeup. You can see that all three of these lanes show a very different protein fraction in them, and that's all from the same species of scorpion. So once you've gotten your fractions, you want to identify their key actions, and this time instead of starting from whole animal and working smaller, start from target tissue and work back outwards. So again, you'd want to study dose response. Again, you'd want to look at what the therapeutic threshold of that might be if you are indeed looking at developing some kind of therapeutic from your venom fraction. So LD50 and LD100 mean lethal dose. So LD50 means the dose required to cause death in half of your subjects. So that's both of those measures are important to know when judging whether you have a therapeutic window. So if your venom doesn't take effect until it causes a lethal dose, that's probably not a great therapeutic candidate in a whole animal anyway. You'll want to look at carefully characterizing your venom. So Edmond degradation breaks it down into amino acid residues, helps you work out what the chain structure of the protein will be. X-ray crystallography will then help you look at what the folded protein is going to look like and judge where the active sites are. And then when you get to the point that you want to begin working with your venom intensively and doing extensive pharmacological research, it's important to think about synthesis. So in the sense of venom, you don't always want to source venom from a wild animal or a housed animal. There can be concerns with animal welfare there. There can be concerns with human safety there. Nobody wants to keep too many king cobras for frequent milking. Some people do. I mean, it's a lifestyle choice. But it's not going to pass as many ethics committees as you might like for the sake of your future therapeutic drug. So at your final stage, when you are starting to think about developing this further, it's important to develop some kind of synthesis for it that is not based on a wild animal. So in assessing your, the things to answer before you continue on is, do the effects that you found scale up to the whole animal? So if you found a really impressive cytotoxic fraction from your given snake, and it holds all sorts of promise as being a future chemotherapeutic because it works really well on cancer cells and kills normal cells much more slowly, you want to find out whether that then scales back up. So after you've worked out that from a cell culture stage, you'll want to then try in a whole animal model. Can it reach the target tissue effectively? Is it metabolized effectively? Is it metabolized in such a way that it can be given by reasonable dosages? Or is this something that you have to give every 15 minutes, for example? And one key thing is target specificity. So if you have a lytic enzymatic venom that goes right for destroying tissues, does it destroy all tissues, or does it just go for the one that you want it to? And that's a really important question to ask. And the more target-specific your compound is, the greater degree of your future success. And again, just to emphasize, you'll want to be able to produce it economically, safely, and with conservation in mind. Here's an example taken from certain, I think this is from coral snakes again, where they have a certain type of metalloproteinase, as well as these three-finger toxins. So something that's very, very general will have an active site that can bind to almost any type of tissue. Whereas you see three-finger toxins, their receptors are much more specific. So they don't act on quite as many tissues. They're much less likely to be to have destructive side effects. Right, so some successful examples. Let's go to snakebite. Obviously, it's a major problem in some parts of the world. There's not really a lot of good options or good outcomes associated with it. Almost everywhere but Australia, you really just, there's nothing for it except to make sure that any clothing around a snakebite is removed so that it doesn't swell too badly. And then you want to get antivenom. That's it. Please don't go sucking snakebite wounds or anything like that. It won't get you anywhere. And the access to a hospital and the access to antivenom are what determines an outcome for people. So that's the bad from snake bites. The good from snake bites is that they've contributed to a lot of fairly key scientific discoveries. So in the case of banded kraits, they're a type of snake related to cobras. They live in, well, this specific species lives in Taiwan. And by the 1950s, neuromuscular junctions were theorized. Acetylcholine had been isolated, but it was poorly characterized and its functions weren't particularly well known. So in the case of banded kraits, bungarus, two scientists in Taiwan noticed that the symptoms of a snake bite from a krait included progressive paralysis, and that included paralysis of the diaphragm, which you use to breathe. And they worked out maybe this has to do something with neuromuscular junctions. So off they went. They found out that these two alpha and beta bungarotoxins inhibited acetylcholine, and they extracted those two key proteins, and then they used them to confirm the existence of acetylcholine receptors, and that was the very first time that that was done. Then the study of anatomical locations of these receptors, we could work out where the density of them was the greatest, where it was the smallest, what was normal. That led to the discovery or explanation of a number of diseases. And in fact, a disease called myasthenia gravis was found to be due to autoimmune destruction of acetylcholine receptors, and the diagnostic test for it is based on bungarotoxin sensitivity. Crotalid snakes, they include scary American rattlesnakes. I really don't like them. Also called pit vipers, distributed worldwide, but the ones in North and South America seem to be particularly prone to being of drug interest. Jararaca is a pit viper found in Brazil, and it's a nighttime ambush predator that immobilizes prey by causing a massive drop in blood pressure very quickly. I've heard a few rumblings in various talks and literature reviews about it having indigenous use by Brazilian tribes who would use it to poison spear tips, but I've never found any publications on that, so take that with your grain of salt. In any case, its peptide component that causes that hypotension has been found to work on the angiotensin-renin loop that contributes to hypertension in people. And it blocks the conversion of a protein which causes the constriction of blood vessels. So that was isolated, synthesized, and brought to market in 1981 with the trade name captopril. So that was the very first angiotensin converting enzyme blocker for people with high blood pressure. It's not used that much anymore because it has a lot of side effects and you need to take it a couple of times a day. But it did lead to an entirely new generation of drugs for hypertension treatment. Other crotalid bites are associated with very serious coagulopathy, and that includes both procoagulant issues and anticoagulant issues and diffuse intervascular coagulation, in which a lot of your clotting factors are used up endogenously, and then you wind up hemorrhaging based on the fact that you don't have any left. So there are a lot of things going on in those venoms, but there are a number of places in the clotting cascade that these venom toxins can work. So at almost every step of this cascade, you can look for a venom component that will act on it in the pit vipers. So some of those hematology drugs there have been delivered or developed from pit vipers, including eptifibatide, tirofiban, and something called reptilase, which promotes clotting. So this one in particular is something that is now used in surgeries where you can draw some blood in advance, develop clots that will be preserved and then applied post-surgery, even in the case of there needing to be blood thinners used in the patient to prevent strokes. But that means that there can be almost a pre-made clot that will integrate perfectly and won't be a problem for the rest of the body or be rejected. The next big thing, I'm fairly confident that most of you would have heard of cone snails. Is that true? Cone snails? Yeah? Okay. Mostly we have that guy there to thank for it. Baldemera Oliveira is from the Philippines, and he is now working at the University of Utah, has been since the 60s. And he worked out that the number of fishermen dying from cone snail stings all seemed to be related to whether the cone snails were fish-eating cone snails. So there's 500 or more species of cone snails all throughout the tropics and subtropics. They've got a few different prey that they target. Some of them eat worms, some of them eat mollusks, some of them eat fish, and the fish-eating ones are the ones that seem to be venomous and deadly to humans. So he wondered whether there would be some vertebrate specificity in the venom, and he found a lot more than just vertebrate specificity. So one example of that is Conus striatus. It's an ambush predator. It causes its prey's muscles to immediately contract throughout the body, and that results in paralysis. So the prey can't struggle and the prey can't escape. And both of those things are pretty good if you're a predator, because it reduces the outcome of your having an injury during your attack of a prey. And that's due to some peptide neurotoxins, which bind to the sodium channels at the axon terminus. That opens those channels, and it allows sodium ions to rush in. A second peptide holds open potassium channels, so depending on how well you remember your nerve transmissions, that does mean that you cause a massive depolarization because the cell can't maintain a difference between the interior and the exterior of the cell. So that means that there's a sustained signal at the synapse, which causes the muscles to remain tensed, and it's impossible for the fish to reverse that. Those ion channels don't exist anywhere else. They exist only in the terminus and in the nodes of Ranvier along the axon. Outside of that, this is a very specific type of channel. It will not work on sodium or potassium channels in any other cells. So if you need to target that exact junction in an animal, that's where you go. You don't need to cause disruption of any other cells. So that's been used to identify where these types of receptors are. It's been used to exclude the possibility of other receptors. So it's not necessarily therapeutic, but it's certainly a useful research tool. Conus geographus is another fish-eating cone snail, and this one does something really neat. So it instead of stinging right away, it essentially gasses its prey. So what it'll do is secrete a type of insulin, which is not the same insulin as it uses endogenously within its own body, but a type of insulin that is specific to fish. So it will push that out into the water. Any fish nearby will become hypoglycemic and drugged and sluggish and unable to swim away, unable to fight back. They will then extend this really weird balloon-like rostrum, start to engulf the fish, and then sting it. And in stinging it, this does cause paralysis as well, but it causes it by the opposite effect. This causes complete flaccid paralysis, where the fish is unable to contract its muscles whatsoever. And that's down to a calcium-blocking neurotoxic peptide called omega-conotoxin. And there's several of them. In geographus, it's called GBIA. There are more omega-conotoxins, which I'll bring up in just a minute. This particular one prevents depolarization of N-type channels. So one specific class of N-type channels, which are only found in pain receptors. Why would you do that? Why would you have something that doesn't cause, that prevents pain only, that is specifically an analgesic agent? There are other ways to cause immobility in your prey, of course, but this one has gone for it by reducing pain. And that means that its prey also doesn't struggle, which is good for it, reduces its chance of injury. So those ion channels, again, only in axon terminals, and they prevent the uptake of pain signals to the central nervous system. So that one reason for that might be what we usually call the Venom's arms race. So there's a continuous cold war that goes on between venomous animals and their prey, where prey are constantly evolving ways to evade the effects of venom and become immune to them, and venomous animals are constantly evolving new ways of envenoming them and causing novel pharmacological outcomes. So the fact that it has this really weird, specific anti-pain compound may be simply down to that arms race that's going on. And then Conus magus. This is the coolest one from our perspective. So cone snails have been touted as the next big anti-pain, everything source of marine drugs that we can think of, but this is the one, this is a success story for the time being. So this also has an omega conotoxin, and this one blocks those channels reversibly. So it's much more possible to be used in therapy rather than as a deadly venom. So in this case, again, it stops the release of pain-causing neurotransmitters, and some of those N-type channels are associated specifically with chronic pain. So not necessarily acute pain that you would get from an immediate injury, but the type of pain that would be associated with cancer or HIV, or old pain due to an older injury that has developed some pathological sensation of pain where there isn't a physical reason for it, but the nerves are still transmitting the signals. And a lot of those sites are bundled at the ganglia on the spinal cord. So that, isolated in the 80s, artificially synthesized and put on the market in 2004 as something that does block pain receptors extremely, opioid receptors. So there's absolutely no interference with morphine. Multiple painkillers for multiple issues. This won't interact in any way with an opioid drug. It also avoids the possibility of tolerance and addiction, and there's no potential for it to be abused. And that makes pain relief available in certain end-of-life care cases and certain cases of neuropathic pain where there's no physical reason for someone to be in pain, and yet they are. So this can wipe that out. There is one major downside to Prialt, which is that it's not a small, the type of molecule is such that it cannot cross the blood-brain barrier, and you can't take it as a tablet, you can't take it as an intravenous injection because the body, first of all, won't transmit it to the target tissue, and second of all, if taken orally, it'll simply be destroyed. So for now, Prialt exists as either a very boutique injection to the base of the spine directly to the cerebrospinal fluid, or in some cases of really bad chronic pain, as a pump that can be implanted to the lower back. So has it replaced morphine? No, not yet. It's not quite the answer to the opioid crisis, but it's a step in the right direction. So in the future, I wanted to point out that you should avoid nonsense. So as cool as venoms of the future sound as our next source of drugs, it gets a lot more press than it gets success. So I think in this talk I've described about 6 venom-based drugs that have successfully reached the market, and that's over the past almost 40 years. 6. So I looked on the internet before we got here to find out whether there was any more being touted, because these do receive some good press, and there's, venoms as drugs is an interesting topic, and it does. There's some nonsense out there. People have used that press to build some snake oil. So cobroxin, nyloxin, those are both so-called cobra venom-derived pain drugs, and you can buy them from, sources of known bullshit. And you can get them as topical gels for your sore muscles and say, like, well, cobras cause neurotoxic outcomes, so this is going to kill my nerves, and that's great. And if you want to do that, then you go right ahead. But you should understand that these have not been evaluated in a peer-reviewed fashion, and they're not under review by the FDA. The other one is Prevagen. I don't know if you have commercials for Prevagen here. But some friends back in the States sent me a video of the commercial of the advertisement for Prevagen. And ostensibly it's because jellyfish are immortal. If you take it, maybe your brain will stay immortal. I don't know. It's supposed to ward off dementia. And we put this up on the commercial up on a screen at a jellyfish conference, and the room laughed and laughed and laughed and laughed. So just because a jellyfish is immortal doesn't mean you can borrow that. So the real pipeline that's out there, there are some cytotoxins from spitting cobras, harking back to that tissue-destructive peptides that are found in hooding and spitting cobras, and those do look like they may be useful as chemotherapeutants. There is some hope for nano-encapsulation of that pre-alt peptide that I mentioned from cone snails. So if that's available, if it can be encapsulated to avoid destruction in the gut, if it can be given a vehicle so that it can cross the blood-brain barrier without having to go in directly to the spinal column, that would also be useful. There are also a few out there that look like they may work for multiple sclerosis or on diabetes. So there are promising compounds out there. It is just very slow. So there's one reason I wanted to bring up marine venoms specifically as being a bit out there, and that's that they're not very well represented in the venoms that have successfully been developed, or even the venoms that have been successfully studied. So 87% of all of the marine toxin databases that we have in terms of both gene sequences and protein sequences, 87% of them are from cone snails. And then that means we're completely neglecting venomous fish, venomous jellyfish, venomous worms, and those are rich, species-rich fauna with plenty of animals in them, with plenty of compounds that they're developing that we could be working on, and we just aren't. And that's largely because of the difficulty in extracting the venoms. So I've talked to you about jellyfish, how difficult it is to obtain pure and repeatable venom from nematocysts. When you're talking about having billions of nematocysts on a tentacle, it's like talking about billions of different fangs all mixed in with mud. So if you can get pure venom out of that, you're doing really well. Fish spines, most venomous fish will have that spine associated with a venom bulb in the skin, derived from the mucus-producing glands in the skin. And this can also be very difficult to get pure or representative venom out of, because as you press on the spine, you'll get a considerable amount of contamination from other mucus sources on the fish. Excuse me. Marine venoms are also very difficult to work with because they're very heat labile, they're sensitive to pH, and they're sensitive to all sorts of, as I say, aggregations and disaggregations of the proteins. They just basically seem to be stickier than terrestrial venoms, which makes them tough. Also not helping matters is the fact that different mature toxin sequences can originate from a single precursor. So you may have the venom be synthesized, and then in post-translation modification, one precursor will be changed into many, many different things depending on the context and the need of the animal. So the way of the future seems to be bioinformatics. So it's possible that the entire procedure I described for obtaining a venom, testing it, breaking it down, testing it in a tissue, and then developing it, that may go the way of the woolly mammoth fairly soon. So the new wave of the future seems to be replacing pharmacological observations with second and third generation high throughput sequencing. And then the combined transcriptomes of venom can be examined without the contaminants. And they can also then be modeled in terms of what they might do without the need to actually physically test them, so that you can carefully choose which compounds to go and test before you go through all the messy work of animal and tissue trials. And that's us. Are there any questions for this or from the earlier talk about plankton, as you like? momento. Tecnología. ¿Quieres probar en inglés o probar en español para Rosa? Oh. Oh, in fibromyalgia. I'm not sure if they have so the only one that's come to market is the Prialt, the ziconotide that has to go into cerebrospinal fluid. So I'm not familiar very well with the disease pathology of fibromyalgia. It's entirely possible. The context that I've most often heard Prialt to be applied in has been in chronic pain or in neuropathic pain, so classic cases of when someone has had an injury and the pain will persist far beyond what's reasonable, to the point that someone is unable to walk or to even touch their feet after, for example, injury, even a sprain. And that's not because it has not healed, it's because the nerve cell signals. So I don't know about disease process of fibromyalgia. Put that into Google Scholar search. I bet you'd find either a paper that discussed why it would not work or why it does, or a case study of where it has worked. Good question.
¿Alguna cosa más? ¿O de la otra ponente? ¿O de algo de esta mañana que se os haya quedado? Bueno, pues vamos a dar...
Muchas gracias.
Gracias por venir esta tarde. Y con esta jornada de bioeconomía azul concluimos todas las que hemos hecho, que contaba esta mañana eran 7, más aparte las que hicimos el 3-4 de mayo. Hemos querido, desde el equipo que ha preparado todas estas jornadas de bioeconomía azul, especialmente enfocadas a biotecnología azul, pues hemos querido presentaros en qué trabaja la biotecnología azul y cuáles son las salidas profesionales ya instaladas en la industria que pueden haber. Todos estos temas y algunos más pues los podéis encontrar en el programa del máster en biotecnología azul aplicada, que el próximo octubre pues lanzaremos desde esta facultad. Así que muchas gracias por venir a todos.